Hi guys, my name is Ezabella. I am a rising junior at the College of Worcester, and I had the opportunity to participate in the HuBMAP program. And today I will be presenting my work I've done during this summer. So my title goes Understanding Angioimmunoblastic T-cell lymphoma in a spatial context using DB-SIG and Codex. A little bit introduction. My name is Isabella, College of Worcester, and I'm studying biochemistry, molecular biology as a major, and I'm a data science minor. So an introduction to a bit of what HuBMAP does. HuBMAP stands for the Human Biomolecular Atlas Program, and its goal is to develop tools to create an open and global platform to map healthy cells in the human body. And it does this by developing new tools and technologies, establishing tissue mapping centers, and collaborating with single cell consortia. So these tools will help us in understanding the relationships between cells and tissue organization. The HuBMAP has already a data that encompasses more than 2,300 data sets, and it spans more than 30 organs. So our next question is, how does the fan lab fit in? So HuBMAP has different categories based on the work that is done under each group, like the HuBMAP Infrastructure, Visualization and Engagement, the Rapid Technology Implementation Group, Tissue Mapping Center, Transformative Technology Development and Demonstration Projects. The Yale University is under the Transformative Technology Development, which focuses on collecting and analyzing tissues. And it also analyzes the effectiveness of innovative tools, techniques, and methods for high resolution mapping of the human body. The Fan Lab has since focused on developing high spatial technology for co-mapping transcriptomes and proteomes using DBIT, which is performed on PFA fixed and FFPE human tissues to generate 3D multi-omics tissue atlas data from the human heart, skin, and kidney. So this diagram is found from the HuBMAP dataset, which shows that the data productivity across many aspects as it shows the funded components, the organ coverage, assay coverage, and the data production life cycle in HuBMAP. So as seen in the image, the data sets which is collected by the lymph node only accounts for 48 data sets. Thus, we decided to use our technology such as Codex, which also was used for only 154 data sets, and DBIT to further understand this lymph node. So a little bit background of the lymph node. The lymph node is a kidney-shaped organ of the lymphatic system, which forms the immune system. It's a secondary lymphoid organ, which is a site for lymphocyte activation, which means when these B cells or T cells are triggered through antigen-specific receptors on their cell surface. And this plays an important role in the body's immune response by filtering harmful substances, and it also contains lymphocytes. And these lymphoma, or when these lymphocytes proliferate and mutate, they are a cause for lymphoma. So more on lymphomas. Lymphomas are a heterogeneous group of malignancies, and they arise from clonal proliferation of B cell, T cell, and natural killer subsets of lymphocytes at different stages of maturation. So they're classified into a broader group called Hodgkin and non-Hodgkin lymphoma based on the presence and absence of Reed-Sternberg cells. Lymphomas are classified based on their cell of origin or from the cells which they arise from. So some lymphomas, such as the AITL, which we focused on, are known to be difficult to diagnose and are aggressive in nature. So further research in this type of lymphoma is important to understand the tumor microenvironment and how the different cellular interactions occur. And we are planning on comparing HuBMAP data sets to further analyze the differences in healthy lymph nodes versus the ones affected by the AITL. So a little bit about AITL, or angioimmunoblastic T-cell lymphoma. It is a rare and aggressive form of peripheral T-cell lymphoma. It originates from 1 to 2% of all non-Hodgkin lymphoma. It is a neoplasm which is characterized by intense inflammatory and immune reactions, as seen on the H&E staining of on my image. And the 2016 WHO classified it as having positive immunostaining for 2 or 3, ideally 3, of the 7 antigens, such as CD10, BCL6, PD1, CXCL13, CXCR5, ICOS, and ZAP. So we can see in the images the immunohistochemistry of 3 of the markers that are positive for AITL. So next is, what is our objectives for this project? So our first objective was to characterize these cell types and the interactions of the tumor microenvironment of AITL. And we want to do this using sequencing-based spatial transcriptomics and proteomics. And we want to also investigate temporal changes that occur within this AITL by using Codex and DAVID-6. And using the healthy lymph node data set from HuBMAP as a reference would help us more understand AITL. So some of the methods we performed were DAVID-6. DAVID-6 is performed by first having an FFPE tissue slide of an AITL patient, and we create the tissue with a cocktail of antibody-derived DNA tags that recognize targets, proteins, and we use a custom-designed PDMS microfluidic chip with 50 parallel microchannels, and we align it with the tissue so that we can further put in the barcodes which we need to place under, so we can then ligate it using ligation linker and an oligo-DT sequence for binding the mRNAs or ADTs. And then we conduct in situ RT to yield cDNAs, which are linked to barcodes A1 to A50. And then we remove the first microfluidic chip, and then we place another microfluidic chip with 50 perpendicular microchannels on the tissue. And then we introduce the second set of barcodes, which contains a ligation linker and a PCR handle. So then we remove the second PDMS chip, and then we image this under an optical microscope or a fluorescence microscope to visualize these pixels. And then we do extract these cDNAs from the tissue sites, and then we incorporate another PCR handle, and then this is performed by template switching. And then we amplify this PCR and prepare the sequencing library through tagmentation, and we perform pair-end sequencing to read spatial barcodes and the cDNA sequences. So, and then further analysis or computational analysis is performed by using CRAT package in R, and also to visualize the data that comes out of these. Our second method that we used is the Codex. So Codex is used to see the protein data from our AITL samples. So then we used an FFPE tissue, which are stained with the antibody panel, and we performed a multi-cycle reaction, which iteratively imaged up to 3 antibodies and a nuclear stain per cycle. And then we chemically strip them and hybridize and re-image for all the antibodies of all the panels. And then we analyze these data computationally using QuPath and RStudio by using our CRAT package. So as a reference, we have here an H&E image of a normal lymph node, and from this, we performed data analysis using, we performed Codex and then performed data analysis, and these were the images we were able to generate. So from this, we can see that this healthy lymph node was positive for certain markers, and then we can also see that there is an organized follicular structure in these healthy lymph node samples. And this is another image which stained positive for the different markers that we had in our panel. This was CD20, which is also a B cell marker, and we can see that they create this organized circular patterns on this lymph node, which we can see later on compared to the AITL, the difference between them. And for this healthy lymph node, there was cell type annotations performed, and we can see there is presence of different cell types on this normal lymph node, including the follicular dendritic cells, monocytes. There was also B cells and T cells of different subsets. And then here we have also another annotation of specific B cell types, and this is the image that was generated. Spatial architecture of the AITL. So basically, we wanted to compare the normal lymph node with an AITL affected lymph node. So the image above shows the normal lymph node as compared to the AITL based on the codex data. And then the bottom is a histological image that shows the respective above codex images. It shows that the normal lymph node as seen in both the codex image and the H&E staining had both a organized circular structures, as I mentioned earlier, and these are the follicles which form from the organization of the B cells and the T cells. But on the AITL lymph node, we were able to see that there was higher proliferation of these T cells, which further showed that these structures were almost unseen in this lymph node. So next on, we have, these are the H&E images of the AITL. We had a total of 30 samples. We collected some as formalin fixed paraform embedded blocks, and we had 23 samples for those. And then we had 7 other samples, and 6 of the samples that were paired with FFPE blocks. So these were the H&E images we obtained from the images that we from the lymph node that we had. So one of the samples that we performed an experiment on was this one, and this is the H&E image of the AITL. And to further understand, this is its codex panel with all the markers that were cell type specific, and it was also specific to AITL. And our next image is a zoomed in region of this box on the codex image, and we zoomed in to see the specific interactions. And so we were able to see DAPI, which is a nuclear stain, and CD20, which was also a B cell marker. CD21 is also seen next, and CD4. We can see that most of the markers for AITL, such as ICOS, PD1, were able to be seen on these markers. And so we can also see that there were tumor cells that were growing on these blood vessels, which were an indicator of one of the characteristics of AITL. This was another image of our H&E stained lymph node that had AITL. And this is the spatial proteomics of this AITL with the different markers as seen in the bottom. And they were stained with either cell type specific markers and also the AITL specific markers such as PD1 and ICOS and CXCL13. And we further performed data analysis by segmenting the cells and using QPath, and then we performed downstream analysis using CRAP package, where we were able to see this spatial clustering of this lymph node. So then we were able to identify there was 14 different clusters for this lymph node, and we were able to generate a HuBMAP plot from this lymph node. And so we also observed this heat map, which also further helped us understand the cell type annotations and also helped us discover which types of cells we wanted to look at. So basically, we were able to identify the different clusters and help them. We used them to annotate for the different cell types. So these were some of our cell type annotations, and we were able to assign a cell type for the different clusters. We were able to identify most using the markers that they were expressing, but some of them, like the clusters 8 to 13 were unknown or they were mixed, so that it was really difficult to understand or distinguish them from other cell types. So as for my discussion, some of the things we've accomplished, or I believe we've progressed a little bit on, was we used codex imaging to further understand or analyze these AITL samples, and it also helped us identify distinct cellular clusters. And so we conducted a thorough cell type annotation, and which leads us to having more cell types and helping us understand the different cell types that are involved in AITL and how these cell types would interact with one another. And so we also mapped the structure of AITL, which ensured that the results were analyzed with our histological findings, and it also confirmed our accuracy for this kind of approach. And so our future works would include to conduct data analysis for DBIT-SIG. We also would like to perform data analysis on the codex data, on the other codex data we have performed. And we also want to focus on these cell-to-cell interactions and also perform these so-called neighborhood analysis. And we want to also integrate codex and DBIT-SIG on the same tissue size, as we were able to use these two different analyzing techniques on adjacent tissues for this purpose. We want to further understand it on the same tissue slides. And so some of my takeaways from my summer is I have learned beyond my scope. As this is a biomedical engineering department, this was a bit out of my understanding, and I learned a lot from just being able to see how these technologies can further help us understand different diseases or different cellular processes. And so I also understood that research is not linear, and there are different ways to approach a problem, and also that we would have to adapt to the different struggles that come on our way, and there are some setbacks as well. And I would love to give my acknowledgement for the HuBMAP program for choosing and for helping me align my interests with my future goals. I would love to thank the Fan Lab for giving me this opportunity. And I would also love to thank my school for providing me resources to apply to these kinds of programs. And last but not least, I would like to thank my mentor, Archie, for giving me the guidance and mentorship that I really needed. Thank you.